Hello, Triple students. We're going to look at monoclonal antibodies today. So I've got some pictures here that link to that. So we've got mice, we've got pregnancy tests, we've got the breast cancer ribbon, we've got babies. Today we're going to talk about this. This links with the trilogy work that you've done on immunity, okay? So let's start with a little bit of language. So we're going to be looking at monoclonal. Now the word mono, so we've got two key words here just to learn. So the word mono means one, okay? The word poly means many. And we know that from like polygons are many-sided shapes. And this always leads me to talk about the game Monopoly. So that is one, many, that's what they mean. That's what it means. And if you think about the game of Monopoly, the aim is for one person to own all of the things. So if a company has a monopoly on a product, then it means that they are the only one selling many of those things. So when we're looking at the word monoclonal, clone means something that is genetically identical. So this means creating one genetically identical antibody. So we're going to talk about what antibodies are just to remind ourselves, but it's really, really important to break down the language in science. So polyclonal means many clones, monoclonal means one clone or one type of clone. So by the end of this, you need to be able to describe the process of making them, what they do, and how they're used, applied into new situations. Okay, so we've gone through plant defenses and human defenses. I'm mostly talking about humans here. So there are two types of defense mechanisms in humans. There are non-specific responses, such as physical barriers, such as your skin and your stomach acid. And they are non-specific because they don't respond just to one type of pathogen. It could be a virus, it could be a bacteria, anything. Your skin is a physical barrier for all pathogens. And phagocytosis, that is where your white blood cells engulf pathogens, so they kind of go and they eat them, okay, and they kill them. That is non-specific, so it doesn't occur just for one type of bacteria or one type of virus, it's any of them. That's the process that happens. So this is non-specific. A specific response is how we deal with specific diseases, specific pathogens, and there are two white blood cells that have that function. There are T lymphocytes, and this helps show the level of infection and the level of response that you have. We're not going to talk about these today, but that's what, if you have the HIV virus and you have AIDS, it's the T lymphocytes that they'll actually have a look at the numbers that you have. Today we're going to focus on B lymphocytes. Now, lymphocyte is literally a cell of your lymph. So these are your white blood cells. So lymphocytes are white blood cells. Now, your white blood cells produce antibodies. Okay, we're going to talk about antibodies more, and more importantly, they can produce monoclonal antibodies. Now, monoclonal antibodies, well, let's talk about antibodies first, shall we? So each virus or pathogen or whatever has these specific markers on them made of protein. So this kind of looks like the coronavirus, so we'll pretend it's the coronavirus. So these would be like markers to show, like a characteristic to show that it is that specific virus. And these are called antigens. Okay, these are called antigens. And when your body is fighting off infection, it will produce antibodies from your white blood cells that will attach onto these antigens. And that helps you defeat the virus. It kind of clumps them together. That means that phagocytosis can happen. And when we are producing monoclonal antibodies, these are specific antibodies that can be used to fight off disease. So, for example, if you have the chickenpox, there will be a specific antibody for the chickenpox, and we could put that in a vaccination. We don't, because it's such a mild disease. But like, for example, MMR vaccine contains 3 monoclonal antibodies. It contains measles, mumps, rubella. 3 antibodies in one vaccination. They will all be monoclonal antibodies specifically for those 3. So there's a couple of videos there. If you look up Twigwell film, I can't show you these now, obviously, but we would do that in the lesson. But worth having a look at what antibodies are and a video of those. So an antibody is something produced by B cells, and it attacks to antigens on foreign cells, forming an antigen antibody complex. So again, if I've got my virus that has got these sticky bits out, these would be my antigens, and my lymphocytes, B lymphocytes, would produce antibodies that are specific. And when they are attached, that is called an antibody antigen complex. It's kind of the same when we talk about enzymes and substrates. So remember, it's my little Pac-Man, this is the active site, and when the substrate is in the active site, it causes an enzyme substrate complex. Specific antibodies are made for specific antigens. So for example, if we have a virus that's got lots of different antigens on it, you would have certain white blood cells that would produce ones that fit that, and then you'd have another white blood cell that fits one that fits on that, and I don't know what shape that would be, but specific antibodies have specific antigens. That's why if you don't have an antigen, an antibody for it, it can take a while. Your body has to manufacture the right antibody for the specific antigen. So a pathogen is likely to have many antigens on its surface. So like I said, you could have some that are like this, depending on how complex they are. And each antigen will cause a different type of white blood cell to clone itself and go, okay, right, I'm in charge of making the ones that cover the round ones. And then you have another one that is responsible for creating, for taking on the triangular ones, etc. So these are all just proteins. If you've got lots of different cells in charge of producing specific antibodies, that means that you can get rid of the pathogen really, really fast. It's when you get a new pathogen where you don't already have the antibody manufactured that it takes a longer time. So let's talk about monoclonal antibodies. So these are antibodies which are cloned from a single group of identical white blood cells. They're all identical in structure and they're made to bind to a specific antigen, so e.g. a cancer cell. So let's say I've got, oops, a daisy, what have I done? Let's say I've got a cancer cell and this cancer cell has specific antigens on its surface to identify it. Okay, remembering cancer cells are usually misshapen. And a monoclonal antibody will be an antibody that we clone that would be a cell that we can produce that is cloned to produce as many antibodies that fit that cancer cell. So this is how we make them. Now this is a flow diagram that you need to know, and I'm going to add on to this worksheet, or you can just draw it yourself. So this is what happens. So we get a mouse and we inject it with a pathogen. So remember, that's a virus, a bacteria, whatever. The mouse's immune system responds. So that means he starts producing antibodies. Okay. So here we've got the specific pathogen with specific antigen being popped into the mouse. We then find the specific B lymphocytes, so the B white blood cells that are producing those antibodies. Here's the antibodies. And then what we do is we get that and we find a tumour cell that divide but don't make antibodies. So there was a lady that her cervical tumour cells were used because they get reproducing. So the good thing about tumour cells is they reproduce. So what we do is we get this antibody producing cell and this tumour cell and we bind them together. So we combine them, we fuse them together and we make a hybridoma. Now hybridoma means of two parts. Okay, doma means, oma means it's a tumour. So it is hybrid tumour essentially. So we've got these antibody forming cells combined with our tumour cells. We check that they've got the specific antibodies that they're made, and then we clone them. So because they're in a tumour cell, they just copy and copy and copy and copy by mitosis. Okay, and then what we can do is we can then collect the antibodies that they're producing. And they're all exactly the same because they came from one cell, and its only job is to produce that antibody. You might want to pause this and make it a little bit more palatable, reduce it down, change it so it looks easy for you. The next slide, I believe, is a blank one, so you might want to see if you can remember it. There we go. Put it into your own words if that helps. Okay, so let's describe the process of making the monoclonal antibodies. So number one, the mouse is exposed to an antigen. Then what happens? The B cells produce the antibodies and they're extracted from the spleen, which is like a blood reservoir essentially. These are mixed with cells that can divide outside the body, like cancer cells. So these are usually tumour cells. And then we fuse them together to make a hybridoma. And they're tested to check that they're the right antibodies. And then the right clone is selected, and then we reproduce it on a large scale and extract it. So this is the important part, is the uses of monoclonal antibodies. So they can target specific cancer cells. So for example, if we have got a specific cancer cell that has this antigen on it, it works as a marker. So we can then produce antibodies as a treatment for that cancer. And then we can combine them with anti-cancer drugs. The drug enters the cancer cells and blocks the chemical signals that causes their growth. So it means that we can actually get hold of them and then deliver a medicine into the cancer cells. That's another thing we can do. So it can either destroy the cancer cell or we can actually stop it reproducing. We can use them to detect illness. So we can use this to detect the HIV virus. And we can use it to detect lots of cancers. So for example, prostate cancer has a specific antigen called a PSA. Okay, so I'm just going to pretend it's that shape. And the levels rise in the blood. So what we can do is, if you take somebody's blood sample and you pop it onto a plate that has the antibodies for PSA, if the blood contains these PSA antibodies, they will bind to that and they will change colour. So it's like a colour strip test. Very much like, just scribbling all over it. Very much like pregnancy tests. So pregnancy tests, so a pregnant woman will produce HGC, and that is found in their urine. And then we can, so when a woman urinates on a pregnancy test, there's this like absorbing strip and the urine goes up the absorbing strip, and then there's like on the outside, so this is the inside, so let's say this is the pregnancy test, there is a window with lines on it. And when the urine goes up here, there is a line of monoclonal antibodies just sitting there, and if the HGC is in the urine, they slot into those antibodies and they turn the line pink or blue or whatever. So it's attached to a coloured bead, so that if the antibodies slot into the antigens, then you know that you are pregnant. If you don't produce HGC, or the antigens in this case, then these aren't going to attach to them, so therefore they won't change colour. We've already spoken about treating cancer. So this is a monoclonal antibody. These are the antigens on the outside of the cell. So they can be used to bind to cancer cells, and once they've bound to them, they attack and destroy the cancer cells. So they can either put something inside the cancer cell, or they can stop the cancer cells dividing. They can attract other cells in the immune system, so your immune system will come and fight it by yourself. Or these antigens can carry, specific drugs that go into the cancer cell and cause it to kill it, die. Okay, I have attached this. This is your plenary, so it's got your task, but it's got a lot of information, so it's worth reading this. And then it says read it in your student book. You can get that on Caboodle, but essentially you've got to do the flow diagram. Have a read of it and do the flow diagram. Please ask me any questions.